So we're group 5 from the Cunha Lab, and our project is Ensemble Learning Algorithms to Predict Scaffold Matrix Attachment Regions. So to introduce our project, scaffold matrix attachment regions, also known as SMARS regions, are essentially regions of the DNA where the human genome connects to the nuclear matrix. They're responsible for structural and functional organization of chromatin into different groups. And SMARS also act as hotspots for retroviral integration, which is the process that retroviruses like HIV use in order to reproduce within a human, which is one of the reasons why understanding more about it is important. Additionally, SMARS are also important to multiple metastatic processes, including the epithelial mesenchymal transition, a process vital to wound healing, but also to metastasization within cancers, which is why researchers need to know more about SMARS. In the past, several computational methods have been developed in order to identify SMARS, but none of these methods actually have accuracy or prediction rates that are usable in the fields of research. And then onto our abstract. So previous studies have gotten us DNA motifs, markers, and sequences that include SMARS, and we collected this in our literature review. But right now, there isn't a complete data set of SMARS in the genome or access to a resource like that. We used R, a statistical analysis classification language, in order to create an ensemble classification model capable of recognizing SMARS within the human genome, and we used SMARS data from the HeLa cell line and HeLa cervical cancer cells, and we used motifs gathered in our literature review. Right now, we use 3 models, XGBoost, Random Forest, and Neighbors. We plan to expand from a classification model to a prediction model, and our future goals are to create a full ensemble model and to expand our data set in order to have results that are more accurate and more realistic.
So onto our research process. So we first started with our literature review, and in our literature review, we read articles on SMARS and also previous packages such as SMARTest. We also looked at the therapeutic benefit of SMARS in cancer patients. Then we branched out to identify two main goals. One of them was data collection, and the other was to identify SMAR motifs. We will touch on the data collection part in a later part of our presentation, and for SMAR motifs, we read papers that identified motifs such as ATTA. After accomplishing these 2 goals, we started on our random forest model. We started with this model to build the necessary machine learning skills for future more complex models. And after finishing our random forest model, we again branched out. We started to research other machine learning models that we could implement, and we also started error analysis and tuning with our random forest model. So we are currently at this stage of our project. We have created other machine learning models besides random forest, which we will highlight later in our presentation. We have also created visualizations and started on documentation. In the future, we hope to create our ensemble model and publish our R package.
For data collection, we were able to obtain the start and end sequences of these SMARS regions from the ENCODE projects, and we collected around 500 of these regions. However, this only gave us the start and the end, so we actually had to use AppScript code on Google Sheets along with the UCS chromosome search tool in order to extract the full sequence. Once we had the full sequence, we were able to create a pipeline to process this data on RStudio, and from there on, we were able to search for motifs such as the origin of replication motifs and things like AT richness and the sequences that you can see on the slides. We also use these following tools. We used RStudio since we coded mainly in R, as well as GitHub to store our code and for others to see our code. In ENCODE, we were able to get our data set, including the start and end sequences, and the UCSC genome browser was particularly useful in extracting the full sequences that we got from ENCODE.
So the first model that we created was a random forest model. A random forest model works by combining multiple decision trees together to improve the accuracy of the model. So it creates a multiple subsets of the original data set by randomly sampling the data set with replacement. This is called bootstrapping. And then after you have all these random samples, a decision tree is made on each subset of the data. So one of the decision trees is shown in the lower right. And the final decision of the model is made from the majority of the outputs of the decision trees. So since this model uses multiple decision trees together, it's less prone to overfitting, which is when it performs well in the training data, but not in new data. Some other benefits of this type of model is that it's less complicated, it's less prone to being sensitive to outliers in the data, and it's also a pretty simple model to code in R, so it was a good model to start with for our project. And then the upper right, the upper right shows the confusion matrix for our model, and that showed that we had an 88.5% accuracy rate when on the test data. Then the next model we made was the k nearest neighbors model. This model works by finding the k closest data points, and in our case, we chose the number 3, and the class of the new data point is determined by the class of the majority of the nearest data points. So the benefits of the k nearest neighbor model is that it's pretty simple to implement, making it a good model for our eventual ensemble model. And also previous literature has shown that this model is often used as a base model for ensemble models. However, the k nearest neighbors model can be computationally expensive for large data sets, but that's not an issue right now as our data set is pretty small. And then here is our confusion matrix, which has an accuracy of 93.3%.
Right, so the last model that we'll talk about is our XGBoost model. XGBoost is a gradient boosting algorithm that combines the predictions of multiple weaker models to produce a stronger prediction. The pros of this is that it has a higher accuracy rate, and it also prevents overfitting, which is when the model basically memorizes the training data instead of identifying patterns. Some cons of it is that it is computationally expensive, but as Sophie said, this isn't really a problem for us right now. So as you can see in the confusion matrix on the left, when we ran our model, we got 100% accuracy rate. Now we suspect that we got such a high accuracy rate on our first run because our data set is currently biased as we have more true data instead of false data. So it's performing better on the test data. So this is a continued source of accuracy for our project.
Okay, next, this is our ROC curve. So the ROC curve, it plots the true positive rate, which is known as the sensitivity, against the false positive rate, which is 1 minus the specificity. So a perfect model would go to the upper left-hand corner, and then the line going diagonally through the center of the graph represents a random guessing.
I'm in a class.
So an accuracy of 50%. So as you can, this type of graph is useful because it helps us understand the trade-off between correctly identifying positives and incorrectly identifying negatives. So from this graph, we can see our models had pretty good performance as we're close to the upper left corner.
Yeah, so in conclusion, we have our main takeaways. With these 3 models of random forest, XGBoost, and KNN, we were able to achieve a relatively low error rate to classify these SMARS regions. And we were able also to compile a collection of motifs which are indicative of the SMARS region. However, one major limitation of our work right now is that we have an unbalanced data set. We have significantly more positive control data than negative control, so we will need to find additional data in the future. This also causes our current models to be prone to bias. So this is another thing we need to fix, as well as high usage of computational memory and processing power. So in the future, of course, we plan to fix our data set, and we also hope to use a bagging or stacking ensemble learning technique with our current base models, along with others such as naive Bayes. And this will combine all the strengths of these base models to create a model which is hopefully provides a higher accuracy rate. And we also hope to apply this model to identify additional SMARS regions. And the significance or application of our research is that this can help other researchers as a tool to carry out their own SMARS-related research. And this can help identify hotspots of retroviral integration.
And then these are our references. And lastly, we have thanks, acknowledgement, and a picture of us at SCUR. Thank you to Mr. Cunha for guiding our research, and thank you to ASDRP for providing the resources and the guidance for this.